Hi everyone. In this video, we're going to look at the process of meiosis. If you haven't watched them yet, I recommend pausing here and watching the videos on the cell cycle and mitosis before you go on to this one. Meiosis is the type of division required to produce the gametes, or reproductive cells. In humans, this means the egg and the sperm, and these are the only cells that are produced in this way. Meiosis produces haploid cells. That is, cells containing only one set of chromosomes, in contrast to mitosis that produces diploid cells, each with two full sets of chromosomes. In meiosis, this reduction in chromosome number is accomplished through two rounds of division. Meiosis 1 to separate the homologous chromosomes, and meiosis 2 to separate the sister chromatids. And before we go into the details of meiosis, let's have a quick look at homologous chromosomes. When we say homologous, you can think of those as being like a matching pair, one of which was inherited from each parent. We can look at the chromosomes by producing a karyotype, an ordered display of all of the chromosomes from a single cell. Now, we would typically use a mitotic metaphase cell for this because the chromosomes will be as condensed as possible at metaphase, and a cell in mitosis will be diploid. So we'll get to see all of the chromosomes instead of just the half that we would get from a cell in meiosis. Let's zoom in to one of these pairs. Each one is a homologous chromosome, and since it's in metaphase, each one would consist of two sister chromatids. You can see the banding pattern is the same on each, as well as the length, the centromere position, and the genes that they carry. Now, while we could see large-scale abnormalities in chromosome structure here, we can't see individual gene mutations or sequences. Now, even though we describe these as a matching pair, they may not actually be identical, as the specific versions of each gene, termed the alleles, can differ from one person to the next, resulting in differences being passed down to the offspring. Consider if there was a gene here for eye color, for example. One of this person's parents may have had blue eyes and the other green, so the homologous chromosomes would still carry the same eye color gene, but different versions of it. So let's have a look at meiosis 1. As we go through, you'll notice that meiosis 1 has some significant differences to mitosis. We'll start in a similar way at prophase 1, with the breakdown of the nuclear membrane and condensation of the chromosomes. But this is where it'll start to change. The homologous chromosomes will pair up, and non-sister chromatids will cross over at regions called chiasmata. Where this crossing over happens, the chromosomes will break and rejoin, switching ends. So, by the time prophase 1 is complete, the sister chromatids are no longer identical. This is one of the main sources of genetic variation in sexual reproduction, and we'll look at another a little later on. The cell will then progress into metaphase 1. But unlike during mitosis, the homologous chromosomes will remain together, lining up as tetrads, or groups of 4 chromatids at the metaphase plate. Anaphase 1 will see the homologous pairs separate, with each one moving to opposite ends of the cell, with its sister chromatids still attached. And this may be followed by the reforming of the nuclear membrane during telophase 1 and decondensation of the chromosomes, although it's worth noting that some organisms skip this stage. Either way, the cytoplasm and cellular contents will then be divided by cytokinesis. Our single diploid parent cell has now been split into 2 haploid daughter cells, each containing one of each homologous pair of chromosomes. Meiosis 2 will look a lot like mitosis, except that we'll be starting with half the number of chromosomes. Both of the daughter cells produced during meiosis 1 will progress through the second round of division, but for simplicity, we're only going to look at one here. We'll start in prophase 2, and again the nuclear membrane will break down and the chromosomes recondense if necessary. As we move into metaphase 2, we'll again see the remaining chromosomes align at the equator. Anaphase 2 will see the separation of the sister chromatids, this time, again, due to shortening of the microtubules. The nuclear membrane will reform in telophase 2, and once again, the cellular contents will divide during cytokinesis. Let's do a quick comparison of mitosis and meiosis to see the different outcomes. Assuming we start with the same parent cell, mitosis will yield two diploid daughter cells, which are identical to each other and the parent cell they came from. Meiosis 1, on the other hand, will produce two haploid daughter cells, which are different from the parent cell and from each other. These cells are haploid because they now only contain one of each of the homologous pairs, giving them a single set of chromosomes, instead of the two sets in a diploid cell. The division of these by meiosis 2 will then result in 4 unique haploid daughter cells. We mentioned earlier that crossing over in prophase 1 is one source of genetic variation in meiosis. A second one is called independent assortment, and this one happens during metaphase 1. Let's assume we have a cell with a diploid number of 4, so it has two pairs of chromosomes, one long pair and one short pair. One of each of these pairs was inherited from each parent, as indicated by the red and blue colors. Now we can have a situation where the blue ones line up on the same side during metaphase 1, producing daughter cells with either both blue or both red chromosomes. But it's equally likely that the long blue chromosome would end up on the same side as the short red one, since the orientation of one pair is independent of the other, resulting in two more chromosome combinations in the daughter cells. Of course, most cells have a lot more than 4 chromosomes, and the more chromosomes you have, the more possible combinations you can produce through independent assortment. In fact, there's a total of 2 to the power of n combinations, where n is the haploid number of the cell. In humans, since we have 23 pairs of chromosomes, this means that there are 8,388,608 combinations of chromosomes possible, just from how they line up during metaphase 1. If you combine this with crossing over, which happens on average 1 to 3 times per chromosome pair, and the fact that any sperm can fertilize any egg, you can hopefully see how meiosis can lead to an almost limitless amount of genetic diversity within a population. Thanks for watching, and if you haven't had a chance to check them out yet, please go ahead and watch the videos on the cell cycle and mitosis.